Okay, next up, Lindsay and co-authors. She's from Montana State and Eric's Boyd's group. She's going to talk to us about subsurface source and biological fate of H2 and hot springs.
All right, thanks for the introduction, Eric. And yeah, I'm a PhD student in the other Eric's lab, and I'm going to talk to you today about the subsurface source and biological fate of hydrogen in hot spring ecosystems. All right, thanks. So we want to study hydrogen metabolisms in hot spring ecosystems so that we can potentially learn more about metabolisms that may have been present in early Earth ecosystems. And so this figure here is showing a depiction of the phylogenetic tree of life with cultivated representatives from all three domains. And overlaid on this tree are the colors which correspond to the maximum growth temperatures of these organisms. So you can see that early evolving bacteria and archaea are thermophilic, and therefore it is likely that the last universal common ancestor was also a thermophile. And so this early life was also likely dependent on chemical forms of energy. So in this plot of hypothesized oxygen concentrations throughout Earth's history, you can see that the origin of life predates oxygenic photosynthesis by about 1 billion years. And so this early life was likely dependent on chemical forms of energy and not on energy derived from photosynthesis. And in particular, hydrogen is suggested to be among the earliest sustained energy sources for living cells, particularly invoked in the hydrogen and carbon dioxide redox couple in hydrothermal systems. And so we want to investigate hydrogen in thermal features for the reasons outlined before, but also because hydrogen is prevalent in thermal environments such as the hot springs of Yellowstone National Park, which we can use as analogs for early Earth ecosystems. And so, in Yellowstone National Park hot springs, the hydrogen concentrations will vary widely, where it can serve as a reductant, as an energy source for the microbial communities in Yellowstone hot springs. And so, when I started to investigate hydrogen in Yellowstone, I investigated it using a suite of geochemical and spatial data. And so this is a map of Yellowstone National Park, should be pretty familiar, and on this map I have plotted the bedrock lithologies as well as the caldera boundaries of the Yellowstone caldera. And so I have mapped here the geothermal regions in red. I've also, using the spatial and gas data that I've got provided by the USGS, I have plotted here all the different hot springs where the size of the dot indicating each hot spring is corresponds to the mole percent concentration of hydrogen. And so you can see that not only do the hydrogen concentrations throughout the park vary, the regions where the hot spring geothermal areas where hydrogen is highest, so these three areas here are all located near the edge of the Yellowstone caldera and in this inner ring fracture zone. And so this brings us to our guiding questions for this study. And the first question that we want to answer is what influences the hydrogen concentrations in the hydrothermal features of Yellowstone, so looking at the sources of hydrogen. This brings us to our second question, where we want to know how do the sources of hydrogen and hydrogen concentrations influence the distribution, abundances, and activities of hydrogenotrophs, or microbes that can metabolize hydrogen, so looking at the eventual biological fate of hydrogen. So, in order to understand the sources of hydrogen, we must first dive into the Yellowstone volcanic and hydrothermal system, not literally, but this is a diagram of the Yellowstone caldera showing the expanse of the caldera, as well as the shallow hot water reservoirs that are fed by meteoric water coming in, which then feed the geysers, hot springs, and other thermal features of the park. Additionally, this diagram shows the magma chamber beneath, about 5 to 10 kilometers beneath the Yellowstone caldera surface, and which interacts with the shallow hot water reservoirs in several ways that I will detail in the next couple slides, and zooming in on this part of the diagram. And so, zooming in on that part, I first want to detail the hypothesized processes of subsurface boiling and phase separation occurring in the Yellowstone subsurface. And so these processes start with the recharge of subsurface water reservoirs by meteoric water. This water, the subsurface water reservoirs then receive gas inputs from the magma chamber with gases such as sulfur dioxide, which when it interacts with water will disproportionate to form sulfate ions as well as hydrogen sulfide gas. And so in addition to receiving these gas inputs from the magma chamber, the subsurface water will also be heated by the magma and boil. And this just causes the fluids to partition into a liquid phase as well as a vapor phase. And so this liquid phase will have the partitioning of non-volatilizing ions such as chloride. And when these liquid phase fluids emerge at the surface of Yellowstone to form thermal features and hot springs, they will result in hot spring waters that are high in chloride but low in sulfate. In contrast, these vapor phase fluids, which contain water vapor or steam, as well as the different gases such as hydrogen sulfide, will undergo different processes. And so this hydrogen sulfide gas will interact with oxygen coming in from the atmosphere in the near surface to produce sulfuric acid. And so this vapor phase and these vapor phase fluids, when they emerge at the surface of Yellowstone to form thermal features, will result in springs that are low in chloride but high in sulfate. So I'm going to come back to these chloride to sulfate ratios later on in the talk. First, I want to go just through some detail on rock interactions as well as gas inputs to subsurface waters. So in addition to sulfur dioxide, the magma chamber will also supply gases such as helium and carbon dioxide to the subsurface water, which will partition into the vapor phase with the steam and the gases. And this highly reactive vapor phase, because it is vapor as opposed to liquid, will be more reactive with the subsurface minerals, as indicated here, which are present in Yellowstone's crust. And so, water vapor within this vapor phase will interact with iron silicate minerals to form hydrogen. And the CO2 gas from the magma, as well as the hydrogen, will react with iron-bearing minerals to form methane. And so, in this vapor phase, which is characterized with low chloride but high sulfate waters, will also have high amounts of methane due to these crustal interactions, and helium, which comes straight from the magma chamber down here. Additionally, these springs will also likely be characterized with high amounts of hydrogen. And so, taking it out of the theoretical and into the real world of Yellowstone National Park, I've plotted here several hundred different springs according to their helium mole percent concentrations and their methane mole percent concentrations. And remember, we're going to use helium as a proxy for the deep gaseous input and the methane as a proxy for crustal interactions. And so, this plot looks especially interesting when I overlay hydrogen concentrations according to the sizes of these bubbles. You can see that springs that have a high amount of helium and a high amount of methane are characterized as well by high amounts of hydrogen. So this leads us to think that high amounts of deep gaseous inputs as well as fluid interactions with the crust will contribute to springs which exhibit elevated concentrations of hydrogen. And so I want to further back that up by looking at those chloride to sulfate ratios I detailed before, which again can be used to determine the amounts of liquid or vapor phase influence. And so if we plot those gas proxies I was going through on the last slide, so on this y-axis helium as the proxy for deep gaseous input, against the chloride to sulfate ratios, and springs with that are more vapor phase influence will plot towards this side of the plot. Springs with liquid phase influence will be on the right side of the plot. And so we can see that springs with a low chloride to sulfate ratio, so a high amount of vapor phase influence, as well as high concentrations of methane, will also have high amounts of hydrogen, as indicated by these sizes of these bubbles again. And this trend holds true for methane as well. And so, this leads us to conclude that these vapor phase fluids, when combined with deep gaseous inputs as well as undergoing high amounts of crustal interactions, will lead to hot springs that are enriched in hydrogen. And so, we want to look at these, the fate of hydrogen in the biology of these hot springs, and to do that we're focusing on this, this hydrothermal area called smoke jumper hot springs, which plots here on both of these plots. And this group of springs is characterized with low chloride to sulfate ratio, so they're vapor phase influenced springs. They have high amounts of helium and high amounts of methane, and they have the second highest concentrations of hydrogen measured as a part of this data set in the park. And so the smoke jumper hot spring area is located here, so one of those high hydrogen areas, and it's located on the edge of the caldera in that inner ring fracture zone, highly reactive rocks. And I would like to thank my collaborators for making the trip up to Smoke Jumper, hike up the caldera, to get these samples. And so we sampled 3 different springs, Smoke Jumper 1, 2, and 3, which do differ in their geochemistry slightly, but you can see from this plot of sulfate concentrations on the y-axis, chloride concentrations on the x-axis, that these three springs all plot kind of in the middle of this log scale of sulfide, but very low on the chloride scale, so pretty much no chloride. This indicates to us that these springs are primarily or almost exclusively vapor phase influenced. And so, because these springs are influenced by this vapor phase, we would expect to see high amounts of hydrogen as well as perhaps methane. So taking a look at dissolved gases measured in these springs, we do see lots of hydrogen. And so smoke jumper 1, 2, and 3 are all characterized by relatively high amounts of hydrogen, with smoke jumper 3 being the obvious winner here, which also exhibits high concentrations of methane when compared to smoke jumper 1 or 2. So taking a look at the biology of the smoke jumper hot springs, here we have the abundances of the 16S rRNA genes with copy numbers plotted on the y-axis, and we're going to use this as a proxy for biomass. And so you can see from this plot that smoke jumper 3 exhibits the highest amount of biomass, especially when compared to smoke jumper 1 or 2. But I not only wanted to quantify biomass, I wanted to quantify active hydrogenotrophic cells, so cells that were capable of conducting hydrogen oxidation. And so to do this, I conducted a serial dilution enrichment experiment on the collected sediments from all three of the smoke jumper springs. I provided these enrichment dilutions with hydrogen as the electron donor, CO2 as the carbon source, and varied the electron acceptor that these hydrogenotrophs were coupling with hydrogen oxidation. So these included oxygen, iron, nitrate, etc. And so if we look at the total numbers of these active hydrogenotrophic cells as plotted on the y-axis, we can see that smoke jumper 3 again exhibits the highest number of these active hydrogenotrophic cells. Additionally, the community of smoke jumper 3 is capable of coupling hydrogen oxidation to a greater diversity of oxidants, so all the oxidants that we provided to it, than either smoke jumper 1 or 2, with 4 and 3 oxidants respectively. So I'm going to focus on this site, smoke jumper 3, for the last couple slides of this talk. And so we threw the kitchen sink of an entire 2x250 Illumina HiSeq lane at the metagenome of smoke jumper 3. We got 108 bins, 68 of which were over 50% complete. And so this is an emergent self-organizing map of all the contigs that bind together, as I will show here. And I just wanted to point out these three organisms that are most highly related organisms present in this metagenome with Acetothermia, Sulfolobus, and also the Cholecellulose, which are these three most abundant organisms, which we can see here in this rank abundance curve of the bins of smoke jumper 3. And so I'm showing here relative abundance of these different taxa, which are present in the metagenome. And so you can see that it's a fairly diverse system in smoke jumper 3, but it's dominated by this acetogen, this Acetothermia bacterium. And so using these metagenome bins, I was also able to look for the presence of a nickel-iron hydrogenase. And so all of the bins you see here in red all have the presence of at least one nickel-iron hydrogenase. And of course, we're looking at the fate of hydrogen in biology, and so we want to see if these are active, if these organisms in this community is actively metabolizing hydrogen. So in order to do that, and this is a work in progress, but we've got this initial data showing through RT-qPCR, so RNA data looking at activities, that we do have some expression, we do have expression of these hydrogenases. And so, just some conclusions from this work so far. The vapor phase fluids combined with elevated subsurface gaseous inputs, which undergo interactions with crustal minerals, will contribute to elevated hydrogen concentrations in Yellowstone National Park hot springs. And so the reactive vapor phase fluids also likely contribute even more to the elevated hydrogen present in these hot springs due to the increased contact between reactants and catalytic minerals in the subsurface. So the hydrogen-rich environment of smoke jumper 3 supports a diverse microbial community, especially within the context of Yellowstone hot springs. And we have this work in progress that show that hydrogenases in this community are actively being expressed. And so hopefully using this suite of geochemical as well as biological data, I've convinced you that subsurface processes do exert strong influence on hydrogen and on hydrogen utilizing microbial communities in Yellowstone hot springs, which can be used as analogs for early Earth ecosystems. And so thanks to my lab, especially my advisor, Dr. Eric Boyd, and everyone who contributed to this work, and our collaborators at NASA Ames, Dr. Troy Fuller and Dr. Kirsten Verstad, as well as all of our funding sources from NASA, especially the Earth and Space Science Fellowship. I don't think I have any time for questions, but thank you.
Well, unfortunately, you're right, we don't have time, but that was really great. And, you know, she will be around, he will be around. So I'm going to pass the baton now to Eric Boyd here.
All right, thanks again, Melody. So it's a